Hi, Dr. Barn. I love me some histology. We're going to talk about cilia. For the learning objectives, I want you to be able to describe the ultrastructure of the three types of cilia and correlate with their functions and locations. Now, in the previous lecture from the microvilli lecture, we talked about apical domain structures, and we had discussed microvilli and stereocilia. In this particular lecture, we're going to focus on cilia. So what are cilia? They're cytoplasmic extensions along the apical cell surface. There are three functional types: modal cilia, which also includes flagella, primary cilia, and nodal cilia. Where are modal cilia located? Modal cilia can be seen in the tracheobronchial tree and oviduct, like the fallopian tube. In the sperm, they have flagella. Histologically, here's an example of a bronchus. Recall again, the epithelium is a pseudostratified epithelium. You see here the cilia bodies, which are hair-like structures protruding out, and you also see this thin dark line, which are the basal bodies. And the basal bodies are basically the anchor of the cilia. Let's look at the ultrastructure in more detail. The axoneme is basically the internal microtubule core, and it's comprised of a 9+2 microtubular configuration. Please know these digits and this organization. Basically, you have two centrally located microtubules, and then you have these doublets, which are 9 sets located in the periphery. Within the doublets, you actually have here's an ultrastructure. You have the two centrally, the 9 doublets, and you can see that there are these small little arms protruding from these doublets, and that is your dynin arm right here. The dynin arms are your key motor proteins, kind of like the motor of the speedboat, which utilizes ATP for movement. You also have these radial spokes and nexin on the side here that actually helps with oscillation and movement of the cilia or flagella. With primary cilia, the second category of cilia, or you can also call it monocilia, these are not modal structures. They can be located in various parts of the body, such as kidney, liver, and retina. Each cell has, again, one cilium. That's why it's called monocilia. Whereas in the modal cilia, you have multiple cilia on the apical aspect of the cell. You have a classic 9+0 microtubular configuration, so the doublets are located periphery, but it's lacking the central two microtubules. They also lack motor-associated proteins. Why? Because they're not modal. So how do they function? With monocilia, they actually respond to fluid flow. So they're fluid flow centers and passively bend with the flow of fluid. They can be chemosensors, osmosensors, or mechanosensors. An example here is seen in the renal tubule, where the flow of fluid can cause movement of the primary cilia, which causes opening of mechanoreceptor calcium channels and the influx of calcium intracellularly. Nodal cilia is the third category of cilia, which occurs during embryonic gastrulation, and activity is near the primitive node, hence the word nodal cilia. It generates left-right asymmetry of internal organs. The structure is your classic 9+0 microtubular configuration. They contain motor proteins such as dynin or kinesin, and the reason why they need motor proteins is they have this active rotational movement, which is like a contradictory. So why do we need to know about cilia and the different types? One clinical correlation is primary ciliary dyskinesia. It's also known as immodal cilia syndrome or Cartagena syndrome. In this condition, you actually have lack of dynin arms. And so you have your microtubular structure, 9+2, but you see that they're lacking these little finger-like dynin arms at the side of each microtubule. You can have various clinical manifestations that can occur in these kids and young adults. They can have frequent recurrent infections, sinusitis, bronchiectasia, which is basically enlargement of the bronchial airways because mucus accumulates within the bronchial airways and can cause enlargement of the bronchi. Situs inversus can also occur. In males, they can be infertile because the sperm is lacking normal flagellar movement. In women, they can have ectopic pregnancy because the oviduct, aka fallopian tube, are lacking normal ciliary movement for the fertilized egg to move within the oviduct down into the endometrial cavity. So just to recap, we've discussed microvilli and stereocilia in the previous lecture. Today we talked about modal cilia, primary and nodal cilia. So do know the comparison and contrast not only of the structures, but of their unique functions. On that note, please contact me if you have any questions or need clarifications, and I will see you at the next lecture. Thank you. Bye.